Almost there.
The plan for that was, I need to.
Okay. All right, I'm going to kick us off here. Thanks, everybody, yet again for coming remotely or here in person. This is the fourth of a four-part series. So we've made it here to the end, which I like to think of as a beginning of new things we can all think about and do together as a community. We've previously talked a bit about how our group and some others do pre-processing. We've talked about structure connectivity. We've talked about functional connectivity. And to me, this is really the crescendo and leading edge now of where a lot of the field has gone, at least in network neuroscience, which is this idea that we have all of these data elements, but we want to make some kind of integrated statement about the data. And in the case of folks predominantly in this room, we probably want to be relating that to something like outcome variables, clinical variables, cognitive functions, or even informing treatments. So in the style of this workshop and series overall, I'm going to take sort of a 30,000-foot view of a lot of issues, but what I'm going to try and do again is provide some narrative clarity and conceptual insight into what's happening in these schemes and try to help us understand what kind of scheme choices would you make to answer specific kinds of questions. And when I first got interested in this area, I built it on a foundation, again, as a clinical neuropsychologist, trying to take 100-year-old knowledge about brain structures and local organizations, how they are grouped together into functional groups or coalitions or modules. And when we think about our fMRI data, this is, again, a surface indirect sampling technique for a lot of interesting underlying activities. When I encountered the functional connectivity area, and I had been first studying closed head injury, it increasingly started to bother me that we were not often talking directly about structural connectivity when trying to interpret functional connections. And to me, this was a somewhat crude but straightforward metaphor where, I look out the window here and I've got a road and the cars are mostly driving that on that and not the sidewalks and through the pedestrians. So there's got to be some sort of structural constraint to the traffic. And I think when you talk to most neuroscientists, we all kind of nod and agree about that. But then on the flip side, we're asking questions like, well, but brains are dynamic and they change their states and they modify in injury and in rehabilitation and disease. And how can we reconcile these two things? When should we look at one or the other or put them together? So today, I want to try to answer a version of all of those questions at once. So I've made this a little bit ambitious in the argument I'll try to make. But I think fundamentally what links a lot of what we're doing is, well, what do we do between these modalities to say something interesting about specific task-based behavior, like I would tend to do in the lab, or clinical outcome measures or interventions where we might do neuropsych assessments, or for us, brain stimulation over here. So what I want to try to do is challenge us to ask, can these structure-function relationships important to cognition, broadly construed, be reduced to simple values? You're going to see, really as a consequence of the field, really complex slides that I've picked from the literature, but what I am going to try and do is point out what you ought to be looking for within those complex schemes for specific functions. And I do think that there's a story here in really the last 15 years of research where people have started at the edge level, those things that are connecting region to region structurally. And then very quickly, after the first few years of that work, they moved into component-based analyses, machine learning, thinking about states from moment to moment. And I want to focus on seeking answers within that space, where by answers, I mean the outcomes that I care about. And so, a few of my brain stimulation colleagues really have, I think, a prudent philosophy to say, well, there's a lot we don't know about stimulating brains to make it move, to make people healthy. But sometimes, like a drug, you can see if it works and then work backwards from there. So I'm going to use a similar line of thinking to say, sometimes you just have behavior and you know something about it works. Let's try to use that to reduce some of the complexity in this space. The one thing I want to say about machine learning and then move on about it is now AI in whatever version you've encountered. This is a long history rooted in what a broader class of tools called statistical learning, which is basically the ontogenesis of where you got all of these fancy neural nets and other techniques. It all boils down to very straightforward statistical principles that get increasingly sophisticated. And so the devil's in the details, and we can only say so much about that today. But I just want to say there's certainly a lot of infrastructure for that these days. All right, but I'm going to begin at the end. I'm going to talk about stroke. I'm going to build up to a couple of specific studies in stroke, not within our group, but in kind of related work that inspired some of our work. And we're in general talking about the problem in the top left. And this problem is that you might have a pair of regions. Maybe you're interested in something like bilateral motor cortex. You have a region in the left hemisphere, the right hemisphere. You can go ahead and use diffusion tractography, like we talked about in talk 2, and build a bundle up and count the streamlines or look at the FA values or whatever your favorite measure is related to your pathophysiology. And then you can look at what we might call normal functional connectivity. And here on the right, I'm going to use this symbolic representation over and over. This is what I would call a univariate analysis, in the sense that you have a single value for what you're calling the structure. Maybe that's how many streamlines are the FA over here. You have some single value for the function, usually, which is maybe you got your correlation coefficient or your coherence. And hopefully by eye, you can see that these two time series between these regions would be highly coherent or highly Pearson correlated. These are very similar signals. And so here we would see, if we did a bunch of connections, we would tend to get a high Pearson's correlation between those properties, the structural connectivity here on the left and the functional connectivity here on the right. And there would be some correspondence between these two features. And I think that this still comprises quite a bit of what you see in the literature and quite a bit of what folks haven't asked about in a given clinical scenario. And so even when I scan across this literature, there's quite a bit of open questions there in my view. Now, in stroke, one of the not nice things for patients, but nice things for a network theorist, is you have this very obvious loss of edges in the structure. You usually may have looked at volumes and locations. As we talked about, you're going to get also massive edge loss, region connection loss, that is. And you might have some corresponding decrease or change in the functional connectivity. And so we're still using the scheme on the top right, and we might ask how that correspondence changes. And the reason you might do that are manyfold. One of them could be, well, you're actually interested in the unique information between the two. You're kind of asking, like, what's uniquely predictive from the function and the structure to your clinical outcome variable. You might be interested in mechanisms about, well, I really thought that the neuroplasticity between M1 between hemispheres was this interhemispheric inhibition process. What happens if I break this edge? Do you see that connectivity go up and down? You can test hypotheses from structure to function that way, and so on. So stroke, I think, is one of those spaces where, or related injuries, where you have kind of frank neuropathology you can see on something like a structural scan. Very straightforward use cases where you can motivate hypotheses like those. Now, somewhere around 2013, 2014, as people were getting the kind of big picture about how these things relate to each other, as we'll talk about, very quickly, certain groups started to see, well, wait a second, it's not just about what's directly connected to each other. You've got brains, they've got hemispheres. You know, what's happening on the left hemisphere may or may not be different from what's happening on the right hemisphere. Those connections can be mediated through the corpus callosum. Any given region could have multiple inputs and outputs, and those things could all moderate the functional connectivity, in part as a function of the structural connectivity, but also the computing that we're doing, which we often can't explicitly see in a scan. So the hard part, I think, in parsing this literature and deciding where to start is basically scaling with the complexity of what I just said. Like, for any given problem, there are a ton of choices you might make about how to study that. And so I'm going to give you a few styles and basically pros and cons of how to frame a few options there. So when I was thinking about organizing how to talk through this from the bottom up, I usually like looking for early and comparative studies. I like to do that because we often have things like corroborating tracer evidence, and we can start to look at very clear features and structure and function, and we can feel like we have a handle on it from direct recordings. So about 14 years ago, Rubinov and Sporns and others were looking at various human and comparative models, principally macaques in this case. And what the game usually looks like is you might have some representation here of a macaque brain on the right. We cut it up into a bunch of regions. So here again is our adjacency matrix. You're going to have a pairwise association matrix where the rows and the columns are regions. And then here, this is just a simple binary structure, right? Is there a present or absent connection between those regions. And what network scientists will often do at this point is reorder the nodes by modular structure, meaning that we use something like modularity optimizing algorithm to ask, how are they grouped together structurally? And you might get some kind of reordering like this, where the algorithm will return a new rank ordering for those elements. You're going to see things grouped together near the diagonal. Here's one big robust group. Yellow is a little smaller, red's a little bigger again. And you're going to notice here, one of the nice things about macaque data is you might actually have directed information. So you'll notice that across the diagonal, there's a few individual differences in the hemispheres going from left to right or right to left or so on. From this, the authors were interested in doing things like, all right, well, we've got the structural backbone to the network. This might be like the information superhighway for the brain. What can we say about the function? Is there any sort of ordered prediction there? And so if you take a functional connectivity matrix, here again, we have measures of correlations among the same pairs of regions. And we can furthermore take any number of those effective connectivity techniques I talked about and try to make guesses about the directed relationships between those regions in the functional data. The first thing that I hope sticks out by eye to you is you actually see a pretty familiar pattern here probably. You see probably 4 main squares. Those are corresponding to 4 modules, and they're in roughly the same configuration as there are in the structure. So one thing I just like to tell everyone out of the gate is the way your structural hierarchy in the brain is organized already says a lot about how you should expect the function to be shaped topologically. There's a correspondence to the overall hierarchy between structure and function. From there, your options are endless. So in this case, in early days for this analysis, people were often thresholding quite a bit. And they still threshold, and it depends on your question. When you threshold the information, interestingly, if you take some of the functional connectivity, you're going to notice that modular organization is quite preserved. Those blocks look more or less the same. They're kind of in the same places as the structure. When you go ahead and you threshold things like the effective connectivity matrix, you might get quite a bit of deviation from that, where here you have one big module that you might be inclined says a lot about the directed relationships in those data, and then the other modules are much more sparse. They're not as well represented there. So I just want to point out to everybody in the room, if you were tracking what I said about functional connectivity, you should immediately be suspicious that many of the early effective connectivity methods were quite deprecated with respect to their ability to recover, quote, true relationships in simulated cases. And so I just want to say as a generalization, there are quite a few new things that could be done, specifically in asking about directed connections from the structure. I'm not really sure that this 14-year-old picture is what we would get these days, and it really is an unexplored area. Nevertheless, if you're not asking about directed connections, we already knew about 14 years ago, function and structure tracking pretty well with each other at this kind of modular level, the big groups of things. Okay. So people started to look at more specific pieces of this. And here was an early study in this space where they applied 2 versions of diffusion tractography. So they did a probabilistic tracking and a form of global tracking, which has a variant that produces more robust estimates at a certain level of the analysis. And encouragingly, even 10 years ago, we saw that, well, if you go ahead and you take correlations between the fMRI and the underlying tractography, you're going to see robust relationships between the two in different types of tractography. So what I'm trying to take away from this particular study, and like all of these, I'm going to kind of emphasize one important feature, is right away people started to see that in the long distance connections involving the default mode network, right? So we've got this posterior cingulate cortex, we've got aspects of the lateral occipital lobes and some of the parietal lobes. Statistically, where the strongest individual connection relationships are between structural and functional connectivity, okay? So right away, people are starting to get this idea that, well, default mode, people are starting to acknowledge that's central to a lot of processes. It's implicated in Alzheimer's and other diseases. And maybe one basis for why that's so important is the structure-function relationship among those regions. And so there was some sense to that at the time. Now, of course, multiple groups got interested in this around the same time when we started to get multiple hands and viewpoints on the elephant in the room. So here was one of my next favorite studies where rather than just looking at each of the individual kind of voxel relationships between structure and function, this was Hermanstad and colleagues about 11 years ago, so kind of synchronous with that other study. We're asking, well, wait a minute, what about extracting all of these edges through a big network? And then can we make any generalizations about structure-function relationships, regardless of whether they make it to thresholds or not? right? And so thresholding was already this sort of, excuse the phrase, but kind of damned if you do, damned if you don't. There's a problem one way or the other, depending on what you're trying to measure. They're kind of looking at a lot of that space. So to boil down some complex plots just to the point, on the top here, I'm showing you that we're going to have two different structural extremes. One of them is where we have two regions physically very close by to one another in the brain. So these are like two nearby regions on a gyrus, for instance, or we could have them farther apart. And they could have more streamlines connecting them or less streamlines. And so quite orderly, basically what this graph is showing to you is that, well, if it's far away and if it has few connections here on the top, there's going to be weaker functional connectivity. If it's spatially close by and has lots of connections anatomically, it has stronger functional connectivity. So that's this log-log plot representation. So interestingly, if you go and look across the hemispheres, basically this plot is showing you that that principle is preserved. So a lot of what is happening in the brain is kind of like local clusters of structures organizing the function. That's a lot of what's strongly connected in your brain. When it's far away, right, those are the things with lots of connections. So that explains the corpus callosum bundles between homotopes in the brain, right? So we've got these like kind of long, robust connections from distant places, including across hemispheres, is one component. And then you're going to get a lot of nearby neighbors through basically U fibers, most likely, that are organizing a lot of that. And that explains this modular story I was telling you earlier, right? That's exactly how the brain organizes, these kind of clustering coefficients in the brain. And a nice feature in this, and you'll see in a few of these studies that I really like, is they just went ahead then and then corroborated this at the single subject level. So they could find this property over and over again when they looked at each matrix individually. So at this point, we've got a few things you can kind of take to the bank, right? Overall, the modular organization of structure explains a lot about function, something like a moderate amount of the variance. Let's call it about 40 to 50%. When you look for effective connectivity relationships, we have some catching up to do in the methods from the early days. When you look at what it is about these connections, nearby and many connections says a lot about function, as does far away and many connections. And then the weakest ones are few connections and far away. Okay, that explains the groupings in the brain, and we can find it in individual people. All right, great. So we've got something we can start to motivate additional and more sophisticated questions. Okay, so we're going to flash forward a few years. And for no particular reason other than I could say a couple specific things that I picked this paper, it's a nice representation of using things like component analysis to make simple claims about complex brain structures, and then relating that to the function and then parsing it out by systems. Okay, so that's the brief part of the story I'll tell you. So we're kind of in the real state up here. People were worrying about, well, yeah, we kind of know this single edge thing. We kind of top out the amount of variance explained between structure and function. Is there anything else we can do about that? And so you have a lot of styles for approaching that problem. In this one, we're using a traditional component analysis with a couple nice features that you can see in this paper. And like many component analyses in brain networks, the first component explains quite a bit about the data, right? And so in the structural connectivity, you've got this dominant component getting over half of the variance in there. And you can go over here and then see what's most loading on that variability. And the way they parsed these systems out, because everybody has their own favorite set of systems when they do network analysis, they have a continuum here between heteromodal systems, those that are supposed to be receiving inputs from many systems and involved in a lot of cognitive operations. We have unimodal systems, only should be getting one. We have, I'm going to forget for a moment the PRI, it'll come back to me. And then we had paralimbic systems. So the first thing they're showing is that there's a heavy loading of these heteromodal systems on the variance in these structural networks. To me, that's kind of reassuring in the sense that when I go and look at things like frontoparietal networks, where the brain varies the most and where there's a lot of cognitive variation are in these heteromodal systems, kind of broadly speaking. There's a lot of exceptions, but there's some pattern there in some network analyses. What's interesting, however, about what they did here then was they took the principal structural variability and then the observed functional connectivity variability. They kind of showed yet again, we have this kind of like modest to low relationship, about 0.43 Pearson's correlation between those elements. But we can go ahead and then look at individual systems. So it just came back to me. So these primary systems, meaning closer to the sensory cortices and insula and other structures, the occipital lobe. So you can kind of think primary sensory regions and motor. There's a very tight relationship between structure and function that goes up to about 0.71 in that case. And then in your heteromodal, paralimbic, and unimodal systems, you see somewhat less. So what does this mean? So this means basically as you get farther away from the primary senses, you've got more opportunity for these functional signals to deviate from the structural expectation, okay? I'm going to get back to that concept in a few ways throughout the rest of the talk. What got me interested in that was I was starting to try to find this compromise between, well, I've got a brain, it may be damaged or not. I kind of want to know how the traffic is supposed to go in general. And it's important to know where the traffic is more or less flexible. If you need to think about a reroute in traffic, you want to use those routes that are the most flexible on that highway system. Maybe the brain works something to that effect when we're learning novel tasks, or we're doing challenging tasks, or when we're trying to rehabilitate into a new function. So around this time and a little before, I had been sort of looking out for that property, and I'm going to keep returning to that in the rest of the talk today. Yeah, please. Any questions, by the way, as I prattle on? Go ahead. Yeah.
So this second function diagram, are you talking here? Are you expecting the people to make natural connection, or are you talking about the direct connection when you say certain function or
Good. So the question is basically, are we kind of like talking about monosynaptic direct connections in the network or polysynaptic multi-steps? You can see examples of both. This one is more that single step hop along the way. And so, yeah, one theme we're going to go through today is what happens when we start to add all of those indirect paths. And like most of this, I don't think all of that is settled. We're closer to settled in healthy groups, but plenty of opportunity in clinical groups. It's underexplored. Yeah. Good question. So the next favorite example I bumped into as I was doing the survey was back to this machine learning concept applied to networks. And this is kind of an interesting area in a few ways. So why would you do this in the first place? So let's think about a scenario. So what this paper was trying to do from Sarwar and colleagues a few years ago is to say, here on the left, we have a structural connectivity matrix. Again, you can probably by now hopefully pick out by eye. You've got one big module here. This is probably one hemisphere. Here's another module here. And then there's a sparser area, which is the interhemispheric connections. So that's kind of a standard way to show a matrix. Each one of those hemispheres has its own hierarchy. You can see little clusters and squares, right? So you immediately can see some modularity there. What they were interested in this paper was to say, well, okay, what if I don't have a preconceived notion about what should predict the function from the structure, right? And so they're trying to basically play the game about, well, I have the ability to estimate some kind of functional connectivity, and then I have the ability to test that, the wrong way around. So this is the real functional connectivity, and then this is the performative functional connectivity, the prediction. And so you can notice right away in this example that if I try to use a neural network trained to the thing on the left to predict some left out data, I can get a pretty decent pattern here on the right. You can actually see this line here, which I just happen to know is the line through the corpus callosum interhemispherically. You can start to see some of the modular architecture within hemispheres is preserved across the two. You can see that some of the off-diagonal blue areas are bluish to some extent here on the right. It's an imperfect representation, but it is a representation of not just the amount, but the configuration of those network features. So let me just say a couple more things about that. So they go through and they nicely share with us a few individual cases of this process. So go ahead, find the structure, generate an estimate, compare it to the predicted data. And the first thing to notice about data, and you're probably going to see this in your connectivity data if you do this, is even if your tractography is working really well, you're going to see all kinds of things in your functional connectivity where you're going to have like big intensity differences sometimes. This one starts to look like it doesn't even know what a hemisphere is. It's just got like everything correlated across them within the hemispheres. When I see things like this in data, just for everybody's benefit, I usually pause and I step back and I check my data processing, right? I start to worry immediately about little hidden motion effects. I start to worry about physiological confounds because it's somewhat rare to expect that the entire brain is interhemispherically having this high intensity correlation. Okay, so nevertheless, when I look at a paper like this, I see that even in those kind of extreme cases, you're starting to pick out some of the relevant information. If we go ahead and plot them directly against one another, again, they're going to see some mean correlation of 0.4 up to 0.7, depending on the case. They did some nice permutation testing here on the right to show that those correlations were above and beyond what would be expected just shuffling the data and the networks around. So at this point, people are continuing to care about, well, really just how much information is there. That's kind of where I classify most of these papers, which is if you're not really deep into network analysis and machine learning and just predicting, it's not going to maybe kind of get you up and motivated to do it yourself. But the thing is, when you think about the relationship in these two coordinate spaces, first of all, the information not explained becomes interesting because that's something about the deviation again from structure, what the function does. And if I can predict this much information just from the structure, well, what should I be able to say actually about my functional outcomes? So in other words, it's kind of inversely motivating caring about the structure because of its shared information with the function. The final thing to say is, well, there are a lot of devils in these details. We really probably want to know, I would argue, especially as people caring about rehab and adaptation, what is it that's not stationary or deviant in these connections relative to that structure? Maybe there's something special going on about what we can't explain. Okay. So a couple years before that particular paper, I had been looking at a lot of these papers, and it really started to bother me that we were doing a lot of sort of correlating across information over time. But when you think about any sampling method, you're getting little slices in time, right? EEG is going to have some temporal resolution. fMRI is going to have some temporal resolution. And you can get vectors to represent, well, what was the quote state, the observable state of that system at that time? Why would you care about that? Well, I was thinking about the problem that every single brain I've ever encountered has some structural backbone. So here's just this toy model projected into 3 dimensions. So we've got maybe one hemisphere on the left, a hemisphere on the right. This would be a very boring brain with only 6 nodes, but it's got its own little baby corpus callosum here. And we were using a technique called graph Fourier analysis, where you can take the concepts that we use in time domain, like EEG, but time is actually just deviation with a single dimension. You can actually think of time as like a little graph, just as a straight line in time. We started to ask, well, wait a second, like, well, if you fold that back on itself and you make it connect to itself, suddenly that can look like a brain graph. So we applied this technique because what it lets you do is ask, well, kind of like frequency analysis, something with no frequency is just going to follow time as a flat line. Something with high frequency is going to go really quick with respect to that line. When you do that to a structural brain network, you can say basically, well, how grouped are these signals based on that structure? And if it was really aligned, you would expect the structure to say pretty much everything about it. Everything would be grouped strongly in these clusters. If it was more liberal, as we termed it there, it would kind of like, ignore the structure. The signals are free to flow in opposition to it. They come and they go. So for me, this was one way to access the question I mentioned previously, which was sometimes the stuff deviates quite a bit from the expectation. As a system, what does that say about things like behavior? So this is my first foray into that space. So what we did was we took white matter networks from each of our persons. We had then from that representation, bold series. So this is when people were doing a cognitive switching task. We took a Navone figure ground task and we turned it into a switching task. Either focus on the big picture, the little picture, and go back and forth in the challenging condition. And what you can do when you have bold data or EEG data or any time series data from brain regions is you can go ahead and just look at what the relative activation of those data were at every single moment you sample them. And we came to find that there were structured states. By structured, I mean they had patterns in them. And some of them looked very much like the bow tie, and other of them were kind of opposing it and not looking much like the bow tie, and everywhere in between. So here's the trick that this analysis allowed us to do. Because the brain network structurally has 2 dimensions, and the state has 1 dimension, meaning just like, here's the measurement I got right now, and I got 1 measurement per region, we can pull information out about what part of that is aligned and what part of that is liberal. Which when I first learned it felt kind of like a magic trick, but it's just like a mathematical way you can use 2 dimensions of information to pull out of 1. When you do that, you actually can take every single subject in your sample, take these states for the subject and their own brain data, and ask about that brain, how much and where are these signals aligned with what I expect, and how much are they liberal? So notice this is very different than what I said before. It's basically saying, I'm going to keep watching the states occur, flowing over time, moment to moment to moment. And in each state, I'm going to be being asked, well, how much of that does the structure tell me? And when it's separate from the structure, my intuition at the time, at least, was that, hey, maybe that's part of how cognitive flexibility works in the brain. It's going to have to deviate quickly from where I thought the structure was going to go to transiently do hard things and to learn new things. So did we find that is the question here. And the answer is yeah, but not in the direction I expected to. And then, you know, like all things, we kind of post hoc made some sense out of it. That's the truth of the matter. So we have on the top here the map of where those regions mostly functionally deviate from the structure. And interestingly, I kind of expected quite a bit of frontoparietal deviation. And it's there. There's kind of like a faint green on some of my favorite circuits, but a lot of it had to do with the insula and the subcortical regions. This makes more sense to me in hindsight because, as we mentioned earlier on, we're usually doing kind of a crummy job sampling these subcortical circuits with diffusion tensor imaging, both because of the resolution of the technique and because of the parcellations we use. So this is something I would like to follow up on, frankly, in the future when we have finer grain parcellations. Because nevertheless, you saw also in the aligned signals, part of those signals are still also tracking with that structure. This seems like a contradiction, but this is a function of having that 2D information from the structure. And if anybody's super interested, we can talk about how the math does that. But what we found here is that actually quite a bit of what the brain is doing in the frontoparietal network is using the structure from moment to moment. Let me explain that back. So what's happening as somebody's going from state to state to state is the prediction about where those signals are going to be as a group is in fact quite well predicted by the underlying structure. So this is not just like correlating the strength of the relationship between pairs of regions. This is saying, given all of the regions together, where would I expect the signals to be? And quite a bit of them are actually just kind of following along with the structure. However, the end of this story is when we went and correlated that with the switch costs, one of our measures for cognitive flexibility, guess where the behavioral relationship was? That was in the flexible signals. So what do I take away from this? A lot of what your brain is doing most of the time has a lot to do with how it's structurally wired up. We can see that in the group. We can see that in the individuals. We can see that in functional connectivity like correlations. We can see it in little state bold vectors like we did here. But depending on the process, you might actually expect to see that variation in the stuff that doesn't track with the structure. This is one of the justifications for doing two things: looking at your functional connectivity, but also understanding how your functional connectivity is informed by the structure or not, because we would not have seen this relationship without this technique. And that was what, to me at the time, was quite compelling about it. Last thing to say, what surprised me. So in addition to the liberality not going in as much the frontal lobes as I expected, this relationship in the flexible signals is in the opposite direction, I thought. I thought being super flexible would be really good for cognitive flexibility. Instead, among the flexible signals, a little bit of structure goes a long way. In other words, as long as the brain isn't completely doing it at random with respect to the structure, being deviated is pretty good for you. And there's kind of a sweet spot in the data, which we could talk about. Okay. So I'll pause here in a second, but the reason that I'm taking this as an example is not just because I know it well, but here's a case where we reduced all of the complexity from the brain data and the functional data through a process that's complex to single number that predicted at 0.57 the behavioral variable that I was interested in. I couldn't get that out of a single voxel. I can't get it out of the structure. I can't get it out of the function alone robustly. I can get it out of the correct way of parsing the two pieces of data together. And I think, again, I don't know how well this generalizes, for instance, to many clinical samples, but this is just the kind of thinking that might be beneficial for some of our data sets. I'm going to stop talking there because I noticed that a lot. Any questions at the moment? Okay. Oh, one more. Yeah, go ahead.
Might be getting to this, but there's sort of in the first two talks, you talked a lot about, or the second, third, you talked a lot about the weaknesses of our ability to estimate functional and structural phenomena.
Yeah.
How does that Venn diagram kind of work in what we can and cannot see in these times now? Talk a little bit here about some sort of discussion.
Yeah. Yeah. Great question. So the question is about, before I was kind of relaying in both structural and functional connectivity methods, we're always wanting for something from the data. They have their major limitations. So when we're kind of now taking the conjunction of the two for structure function, is there a new problem there? My answer is probably yes and no. So you're not doing anything better than either technique alone, in the sense that you can't make the bold signal measure things that it doesn't, and you can't get around the diffusion tractography reliability validity issue. What you perhaps can do is, when you're looking at the complexity of the data as a group, there could be structure there that you don't see in any one of the elements. That's basically how these component in this graph Fourier thing I mentioned, et cetera, tend to find meaningful variance in the data. In my view, the best stress test for everything in here, and I'm going to say today, is novel left out data where you don't get tricked by like the covariance assumptions. I just think so much of neuroscience right now has that problem fundamentally, where you just don't know where the reliability is and isn't until you test it in the hardest cases. I guess that might be a general response. Great question. So again, if someone wants to talk more about this specifically, I still think there's a lot of promise here, and it's probably worth talking about. Oh my goodness, look at all this stuff. All right, so we're not going to go through every element. What I'm going to do here is point out that there's an entire arc of science, I think, that doesn't really clearly penetrate a lot of clinical areas. And this is one of several ways of doing it. So you have, I think, people more like me who started out on the clinical end of training and start to get involved in neuroimaging, and they get to kind of like a certain level of depth with that, frankly, out of training and time and energy, I think is the biggest variable. You have then a whole other branch of folks, and these people become more numerous really every day, are people with engineering and computer science and physics training backgrounds. And they're kind of looking around and saying, hey, you neuroscientists, you didn't think about all of these physical things when you're trying to study your problem, and we're going to go and solve it. I'm being a little bit cynical about that. This is my favorite example, actually, about like, let's please start actually finding ways to make people work together. Okay, so there's this whole way of thinking about the brain as a coupled system. And by a coupled system in physics, what I mean is literally just like things that are networks that have things attaching them. So those folks look at a brain, and to me, it's a salivating problem, right? It's like, great, this is the penultimate coupled system. And what we're going to do is we're going to cut it up with our parcellations, and then we're going to use our DTI image to inform that. And then now we're going to try and do things like extract networks. We might be doing something like actually explicitly model something like an oscillator, because we think the brain might look like a coupled oscillator system. Maybe we can go ahead and then extract resting state networks we knew about from that and tie all of this together in a structural functional relationship. And in this space, what people tend to measure are features like metastability, which is the idea that the brain can occupy certain plateaus over time in many different ways. You can pull out the eigenmodes of the correlations among these things. So now we're getting even more abstract through the data. What I mean by that is I can look at the correspondence between structure and function and then look for patterns in that grouping of relationships. So we keep getting kind of more abstract away from the data elements in this work. But what that allows the folks in this domain to do is say, well, that allows me to look at dynamics of the system in a couple of novel ways and see if they're related to things like cognition. So I picked this one because they actually related it to disorders of consciousness. And so what you'll see in my toy model up here is I'll give you a little extra fun green arrow out to what we're trying to relate these things to. So we're starting to think about behavior, clinical samples. And so we can have here healthy controls on the left. We're going to have folks with the most severe conscious impairments on the right, and then a group that was in between. And all they're showing here in a nutshell is that, well, this kind of correlation space between the structure and the function is actually more collapsed, meaning more glued together in the disorders of consciousness. Okay. And there's a lot going on here, but basically they're showing different components of that same property. Why am I bringing that up? To me, it's really interesting about brain networks that when you start to look at how well these things are glued together or not, you very quickly see across groups that it's a property that it's kind of a little bit mushier, right? There's enough of a separation in each of our healthy brains between the structure and function to facilitate adaptation to tasks and to facilitate learning and to facilitate neurorehabilitative recovery. As you start damaging brains in various ways and taking away cognitive states, it starts to get a little bit less flexible with respect to that structure. It sticks more like glue to it, in other words, is a way to think about it. So it's not always good for your brain to be kind of, stuck in its ways dynamically. You actually want it to be flexible off of that backbone. We see that here again in disorders of consciousness. They went ahead and actually also showed here on the bottom row is that in each of the samples, you can take that eigenmode, that big component of structure-function correspondence, and that also explains a lot about how the brain dynamically navigates over time in this property called metastability. I'm bringing that property up because you may see that term somewhat commonly in papers like this, and various groups now have associated this way of brain states navigating a space to clinical outcomes and clinical groups as an overall way of talking about the way brains navigate as a system. So I would just say in general, it takes a few steps to get to this property called metastability, but it doesn't need to be measured this way. You can actually measure metastability in a relatively straightforward fashion on much less complex processing schemes to look for things like, sensitivity in your groups or sensitivity to the outcome. I've seen enough of these now that I think I'm comfortable recommending it's worth looking at in data if you have network data to look at it in. It seems to be responsive to a lot of disorders, and especially to disorders where you have large changes in cognitive state like loss of consciousness or large changes in the structure like strokes, for instance. Okay. So let's go way back down to something that I think is a lot easier to think and talk about in general. Let's go back to stroke. Let's pick it up where I began, which is to think about kind of the simplest case scenario for structure function damage. And so what we basically are talking about is a problem where you're interested in structure, you had your diffusion measures, you're interested in function, you had your fMRI measures, and then you blew up something in the structure that you can observe. And if you believe that there's some correspondence between the two, that's basically the Griffiths papers we'll talk about, which is that we're going to disturb something about it, and maybe that says something about behavior. And so in this study, there's a lovely paper. It's a few years old now, but if you're kind of a first timer to structure, function, and stroke, this is a good place to start, in my opinion. And they're, again, doing a latent variable mapping. So they're picking out the overall structural variation in these networks, and then they're trying to look at the relationship between these structural disconnections, these little strokes to the edges, and map them out in the brain and then use them to relate to behavior. And so what they were showing here is that if you look within and between networks, and you look within and, between and then within hemispheres, you can see that the intrahemispheric ratios of the strength of these components shifts in response to stroke. What do I mean by that? I mean that this gobbledygook here on the bottom right, this big cluster of network edges, moves as a unit in response to stroke. I can have something about an interhemispheric effect where if I'm going to have lesions in a sample, I'm going to have a whole bunch of edges interhemispherically that tend to fall out as a group because I had a big stroke and it knocked out a lot of edges in that group. I'm going to see corresponding to that structural change, groups of functional changes, okay? So what was kind of clever about this paper was they were saying, well, when I disconnect the hemispheres physically, I'm going to expect a couple of things. I'm probably going to expect interhemispherically the connectivity to change, but I'm also going to be interested in what changes within those hemispheres. And so what they showed in this paper is just pulling out that basic idea, you can see a huge drop in the interhemispheric synchronization in the brain. That's kind of straightforward. I cut the stuff. It's probably less synchronized. That's violating the property from the Hermannstadt paper back in 2013, right? Long, strong distance connections get a lot of variance. That drops away. Interestingly, they were also showing that there were a lot of these upregulated connections within hemispheres and between hemispheres. Within hemisphere, you had a lot of this anterior posterior recruitment of like demand responsive and attention networks, stuff that I like to spend a lot of my time in. And then between the hemispheres, you had a lot of these kind of interesting connections between non-homotopic regions. Why am I bringing this up? I think in my kind of like, first order way of thinking about neurorehab syndromes and recruitment is the homotypes want to tell me a lot. What this analysis tells me is that if I knock out the opportunity to see the homotopes, I've got some other kind of process happening around it that involves fundamentally two things. I've got a bunch of recruitment about those kind of low detention demanded networks, and I've got a lot of recruitment about these sort of slantwise, less predictable relationships among other regions. This component is really interesting to me. This remains interesting. We talked last time and before about concepts like bypasses, indirect connections through these syndromes. And so to me, there's some meat in these first two, which is kind of entirely unsurprising in a good way. And then there's a whole bunch of this, well, what the heck is this other stuff and what is it doing for us? Okay. So what they were showing at that point was that we can get a lot of these relationships between the factor scores that we pull out and the overall interhemispheric, intrahemispheric, default mode, attention network relationship. So there's a kind of basic relationship between dropping out this big component and these other pieces of connectivity going up and down. are they all going to be 0.92, 0.63? How much of this was only searching for certain things? How much was their data set? I have a lot of open questions about that personally. But nevertheless, we see these components shift and then the other connections come online or offline the way I talked about. When they look at the behavior, they could find that examining the structural disconnections and the functional connectivity got, depends how you squint at it, but kind of a reasonably similar level of covariance in the data, meaning in the language domain, you could see that, dropping these connections and dropping structural and functional connections was similar and so on through attention, motor, memory, and so on. So what I take away from this study is what I mentioned about the organization, tension kicking in, and there's a lot of extra bypass stuff, what's going on with that. And we're still kind of living in this 0.4, 16% of the variance at best land when predicting them. Interestingly, when I had done my simulated attack paper with Peter Turkeltaub, we got structural connections alone somewhere in like the 20% range of the variance when we tried to use that overall simulation component to look at the behavior. So how do we do better than this? What else can we look for was what I was thinking. And I think clearly Griffiths and colleagues thought about it. This is basically the bridge between what Griffiths did and what we did with Brian Erickson in our bypass paper. This is part of the inspiration for that work. So I think Griffiths saw that finding.
Can I interrupt?
Oh, yes, please, Laurel. I'm going to keep going until you interrupt, so please do.
Yeah. Can you go back one? Just
Yeah,
on
please.
the lower right. I'm just curious, the adjusted for lesion volume. Does it look like the functional connectivity was a better predictor in some cases there? The red looks a little, the partial correlation looks bigger for the functional connectivity than the structural.
Yep.
I wonder if you could comment on that, because, sometimes one of the problems that we have is, where do we start? Well, what's the bet? Clearly if we can do both, that would be great, but what are we going to look at first? And I think we'd asked before, what do you favor? And you said you lean towards structural. Here, this looks maybe like functional might be a better predictor.
I'm glad you brought that. Yeah, because I can answer that directly. So I think it depends what you look at, unfortunately, is where I have to say. I think if I buy my own simulated attack results, what I think I take away is that there's an overall mass effect on the lesion that is not entirely just about the lesion volume. That is then expressed through these kind of like larger overall topology measures of the brain, right? So if I think about the pattern of how everything is configured together, that's why we get some sensitivity and modularity and efficiency, which is not represented in this analysis from Griffiths, right? That's about the overall way the elements shake out. That's probably what's making the behavioral variance higher in that analysis. I completely think that I'll take away from the Griffiths paper is for me, I'm looking at the attention one and I'm thinking immediately about that recruitment of those really flexible systems, right? And on the one hand, some part of that signal I know is predicted by structure, but this is also telling me it's also right to think about not all of it. So I'm kind of like a boring linearist about this, right? It's sort of like you thought your lesion volume mattered, okay? The structural connectivity is going to probably say something more on top of that. The functional connectivity is probably going to say something more on top of that. So I'm a big fan of kind of stacking the analyses in that direction. So then as you add each piece, there's something, coherent to say about it.
Okay.
Yeah, that makes sense.
Thank you.
You're welcome. And again, I'll just remind, I am still really concerned, and I think that's the right strong word, that there's enough differences in the basic processing routines and partitions and how everybody does these studies. I am not clear at all how much I should really expect like that attention score to look the same if you were to run it through my pipeline. So I think we still have a big problem in the field to ask questions like that. Yeah. So again, that was part of why in our lab, we started let's mine several versions of the network because we can then ask across those domains. Okay, and we're almost done here, and then yes, please do interrupt with anything for anybody that comes to mind. So I think Griffiths, we're kind of responding the way a lot of people did, which was, you're kind of really focusing on the direct connection problem in the last paper. Up here in my toy model, well, wait a minute, the stroke is going to intersect a lot of things that interact with a lot of other things. So this idea that there are multiple pathways for many pair of regions in the brain is to me one of the most compelling things about network neuroscience approaches, right? Is that we all probably believe most processes have some kind of multisynaptic process, whether you can measure it or not. The direct connection probably says a lot. We already talked earlier about that, says a lot about the function, but not everything. And so what Griffiths was doing here was starting to look at, all right, you kind of have this direct structural disconnection, which is if A and D were really connected and I blew it up, maybe some kind of bypass goes through C to D. Okay, so that's a novel path along the network. And then more problematically, you can have indirect structural connections, meaning, well, maybe I already had A to C to D, and then I blew up part of that, and then it compromised that path, and then it had to go somewhere else. So I think there's something about if you believe that brain regions can talk to one another past one node, one hop in the network, it's probably motivated to ask questions like this. And I think that this paper opened that topic up. So again, there's a lot of sophistication in this paper that I could take a while to dig in. We could journal club that. But I think in a nutshell, what we're kind of looking at is positive and negative connections. And then when you start asking about these spared and disconnected regions, you can see different trajectories in where the function goes. And so this was the main innovation in that space. So basically, what they were showing is that if you've got differences in functional connectivity here all the way on the right, I'm looking at now, this is, the structural connections are spared here. We've got two types of disconnections, and we tend to see that the direct disconnections gives you the more prominent decrease in functional scores, and then there's some residual in the indirect connections in this direction. So this is for the initially positive connections, things that were strongly positively coupled. And then you basically see some opposite here, and then they were kind of finding it dubious that the negative connections were statistically different, and the effect was overall weaker in these data. What do I take away from that? So one thing we can take away from that is here again, I found it reassuring that direct connections say a lot, right? Nearby, strong direct connections still say a lot about functional connectivity, reassuring. I don't have to reinvent 10 years of network neuroscience. Indirect connectivity may say more about things that are positively coupled with one another. So I kind of think of that as like, if I'm trying to get to my neighbor, but I have to pass it, down, like whisper down the lane style, it's going to be better to get messages that activate downstream. These more nearby neighbors and negative connections, meaning things that suppress one another, track a little bit less directly with these direct and indirect connections. In hindsight, I have, a couple kind of low-level questions about this study. I'm a little bit worried that, we've had this huge debate over grand mean scaling and functional connectivity. There's a lot of things about how you process the time courses that make things look more or less positive. Nevertheless, it could be that if you're trying to do the structure function game, you might start with the positive connections about kind of like what adaptively synchronizes or not in your data. That may be one place to start there. All right, I'm going to pause there. Anybody have questions about this study? I'm going to mention we didn't do this follow-up yet, but it's baked into hopefully our more constrained version of this. So Brian Erickson and I had published this bypass paper. We found there was sort of interesting recruitment of, in some cases, frontal parietal networks or deeper brain structures in predicting the outcomes just from like core swab measures. And one of the things, of course, I keep finding over and over, which we've talked about, is like, behaviors sometimes are a lot more specific than just throwing it all in the hopper and asking about the network question. So we're anchoring a lot of that to the role of the IFG in language and language control. And what we're curious about is if I already have like a hypothesis trained to the idea that IFG mediates the process, do we actually see something like stronger coupling between the recruitment from that anatomy to the function, right? Because that to me would demonstrate that there's actually active circuit recruitment to an extent. So I think that you can always take this like super global way of attacking the problems and then use your neuroscience training, use your domain knowledge and say, look, I really think these 4 nodes are the circuit. Okay, great. Like zoom in on that and test that explicitly. Even if you think you're going to get more variance from the big connectome version, I would still think it strengthens your science agenda to zoom in on that problem. And I don't think we're seeing enough of that yet. I mean, I see this branch causal across the street. We get better signal when we go and do that in the network analysis, right? So this is probably a truism of studying brain circuits to an extent. So I need to make you aware of one last thing, including a new development for us. I just want to, well, hopefully go somewhere that I'll mention. There's a whole extra industry we didn't talk about simulating oscillators, like I mentioned. So the virtual brain back with Jirsa and Deco was now 14 years old or so. And this was the idea of take a network, couple oscillators on it and look for those oscillations. And you can use oscillator predictive math and make inferences about how that system might work. And it turns out, again, those have weak to modest correlations with various cognitive and clinical outcome measures. So that's kind of cool. There's another entire branch of science, which is where I started out partway through postdoc, looking at control states and control roles, meaning we kind of like abstract away a lot of details about how the dynamics might work. And then we ask about, well, how might the brain transition through state to state? And then can we intelligently tailor input, something like brain stimulation, to make it better? So I'm having a conversation with Rick Betzel, who's currently at Indiana, to see if in one of our personalized brain stimulation studies where we found an effect of cognitive flexibility, can we basically kind of validate some of these concepts? They've come a long way in the last few years, and it might be time to ask that question. The last thing I want to say in general is we have, I'm really, as a psychologist, I don't like using the word crisis these days because psychologists use it too often, but it's like a quasi crisis to me that we all kind of sit here know. One of the folks asked in a previous talk, like, well, like it's nonlinear and it's kind of chaotic and state means lots of things. So how do we do that? I think one of the biggest areas that we still have to solve in neuroscience is the nodes are not inert, homogeneous things. They're computing, they're doing things internally, they have internal states and representations. And what gets passed from node to node and how is in some domains actually pretty well mapped. So you can use that to constrain, again, how you try to model these networks and dynamics. Or if it's not mapped, you might have a hypothesis about how it could work you can test and these network data. And so like there's little pockets of this science all over the place that I think are ripe for the taking for clinical and applied communities or basic science communities and clinical pathology that I just wanted to mention here. So here are my takeaways, then I'll stop and we can chat about anything on your mind. From this whole series, I think you got to talk to people. I hope you like talking to people. I do. And I think that it's the domain neuroscientist, network scientist interaction that really moves us forward. I think we have too many silos even in 2024. Do not forget your strengths and field foundations. I might have knocked out the slide here. Sorry, I'll get it back. And by field foundations, I just mean don't forget what you think you knew from 100 or 150 years of neuroscience. And don't think that what you learned about spike train models, last week might not be relevant. It probably is to how you're going to construe these questions in brain data. I hope that I've compelled you that structure and function are somehow fundamentally linked, at least at the level of neuroimaging. I think that that's not even a controversial statement to this community anymore. Like clearly there are relationships there and we keep learning something about them. But I also want you to think about this as a clinical systems problem, meaning like it's intelligent to ask, what should I say about the structure? How far does that get me? When does the function deviate? How much does that get me, right? And kind of work backwards from that logic to help frame the problems. And last, I like me some networks. That's why I've talked about it for 4 series now, is decide if it's a network problem or not. It might not be. And I think I'm worried a little bit sometimes about, we all have this like compulsion and grant driven drive of like, get the next technique in there. When you actually get down to doing the science, it might not be the thing to do for you. It may be a much different method that answers your question. However, obviously, I think that for many of our questions, they can be enriched by network analysis. And again, I've been grateful to Laurel and Branch for motivating this little series, and I'm hoping we can talk more about it. All right, so with that, thanks everybody very much, and I'll be here for a bit for any more questions we have. Oh, thank you. The Drexel people are being too nice to me, but I appreciate it. If not, can I get somebody non-Laurel and then Laurel? I know she and I are both waiting to get somebody else to jump in. Do we have a brave, unwitting volunteer? All right, Laurel, go ahead. Maybe you can stir the pot for us.
Well, I'm actually going to take it up to a much broader level, John.
Yeah.
So thank you for these very sophisticated talks. And even though I definitely didn't understand everything, I feel like I have a somewhat of a better overview thanks to your really well-organized presentations. But so I think what happens in the behavioral world, the cognitive world, or even the rehab world, is people are wanting kind of a magic bullet or wanting a biomarker, of some problem. very much more simple than what you're talking about. And in the motor domain, cortical spinal tract integrity has gotten people pretty far. So people are starting to look at, doing good old T1 weighted imaging, and then, inferring the amount of disconnection of the cortical spinal tract, and then looking at hemiparesis. And not surprisingly, it predicts like incredibly strongly. That's kind of almost all you need to know. Then also whether you get an MEP when you do PMS. That those two things can tell you kind of everything you need to know about damage, about functional deficit, and about recovery. So I guess what I'm wondering, the reason I said all that was it as an example of where you can know kind of one simple thing and it tells you a whole lot. Do you see any domain within cognition in which you feel like there's, other than motor, where you feel like there may be a similar prayer of moving us toward that biomarker kind of answer toward some of these complex relationships?
Yeah, and I actually think we encouragingly see this, and there are hints all over these more complex things that I've seen where, like, the scenario you described to me is part of why you expect from like sensory motor domains this tight coupling of signals, and you don't really have to even worry that much more in the imaging data. And I actually get suspicious when those correlations get higher just because of the reliability of the data. So that to me is actually reassuring that, you don't have to make it harder than it is. Like maybe what we need to do instead is worry more about mechanisms within that specific circuit. How do we push it around? Like, really just zoom in on that and not worry so much about the stuff around it. For my worry, it's just a worry. My worry is when you get way out there to the kind of systems I spent more of my time in, like frontoparietal circuits and executive control. I would argue we still have a measurement problem. We often don't entirely know what we're trying to get out of those systems. And I just worry they are going to require more sophisticated kind of dynamic models and assumptions about how they work. That's what it looks like in the imaging data. When you try to zap it with TMS, like it's a little squirrely and harder to predict. So that's kind of my general gradient. I think where it's simple, I think things that get mediated through subcortical systems with well-defined a priori pathways you can point to, great. Start there, build out from there. I think if hey, it's this nucleus, down in the thalamus, like go there, right? Like try to challenge the imaging to get better at imaging that. So that's kind of why, as a practical matter, I'm right there with you. Like if you have that, man, like you mine that for everything you can, you work hard on it. And then when you need these other things, that's when you start looking, in my opinion. And I like how even the way your question frames it tells me it's a similar line of thought, right? Like, I have to motivate, it's getting me more than the previous thing. And I myself, even through exploring them, have come full circle there. There's too many cases I can tell you about behind the scenes where I'm like, man, it doesn't look like this is going to help me help people. Right.
Yeah. And I mean, the data you showed again, reiterated that structure function coupling is going to be a lot higher in those sort of primary sensory motor cortices, whereas as you get them to more complex functions, relationships get. And so that's really actually helpful in sort of predicting what kinds of behavioral tasks could you expect to have very simplistic biomarkers versus those that are going to need more complex models. So that's really useful. Yeah. I'm going to wait because I do have one more question, but anyone else, please?
And I'm glad you do. Don't, like, please ask them. But yeah, anybody else want to jump in? I have a group thought question because we have a few minutes. I mean, so I think Laurel and I feel a little like-minded about kind of what gets you up in the morning to look at which part of this potentially. When I started this, it's exciting. I like sophisticated methods, right? I like understanding how they work. I like thinking about complex brain functions. But I guess I have a question for the group, which is, do we all think that progress looks like boiling it down to as simple a principle as possible? Or do people worry that we're actually going to need just really complex things at a certain level to solve other things? Like what's the hope quotient in the room, right? For like, just get the simple thing and move on. And I'm interested in any views on that.
I mean, sorry, go ahead, Aaron.
Nope, you can go.
All right, I'll
Um,
moderate. Yeah, Amy, then Aaron, go ahead.
yeah, I mean, obviously, you think Occam's razor, you want things to be as simple as possible, ideally. But I do think you're right, John, that we're just realistically dealing with very complex issues in a system that has, enough complexity, sorry to be redundant, where it's just I think to really get the full picture, we do need to embrace some of these more complicated methods. But that's just my feeling.
I think Aaron was next up there.
Yeah, I guess I was gonna sort of flip the question around a little bit, which is, at some level, you can find a way to pull anything out of anything you want, right? And so I guess what I was wondering is, at what point should we stop and say, okay, our methods are now manufacturing results that aren't really there?
I have my thoughts there, I think, but yeah, what are others' concerns in that space?
Well, I'm always looking for the best predictor of a dependent measure. So, I do think it seems to me that I don't really understand, Aaron, your question. So like, let's say you're building these stacked predictors, like what John was talking about. You start with lesion volume, then you add structural, connectivity, then you add functional connectivity, and you get in the end a really wonderful prediction of the behavior. What's
Well,
manufactured about that?
John was talking about how you can sort of go into higher and higher and more abstract levels of things like metastability and these other measures,
Right, true,
And at some point, you're probably just quantifying noise, right? And so the question is, where do you stop?
Yeah, but that's why the behavior becomes critical in my mind. Maybe I'm
But I think
naive, but
you can find a correlation with anything you want, including behaviors, if you go far enough into the noise.
You mean statistical norms?
Yeah.
Yeah,
Like
yeah.
you can pull out anything you want out of any data
true,
set.
right. Well, isn't that a thresholding issue then?
Well, I guess the question, so, the question is like, how far should we go with these methods?
I do worry, and I think that's a great question. I think there's a few ways to answer it. I'm worried a little bit about what I think we saw in just fMRI research not too long ago, which was, one of the favorite papers for some of us was the paper that was.
What? Oh,
Oh, you're frozen. Oh.
shit.
Is that on our end or is that on their end?
Did anyone else?
Can you hear us, anybody?
Yeah, I can hear you. I think it's on John's end.
Yeah, I think
Okay.
so too.
I think they changed the title later, but Voodoo Corp. There's only so much.
You froze for John.
Oh, sorry. Where did you lose me?
John, you froze for a really long time.
Oh no, am I back?
Sorry, yeah.
So I'll try to say it faster this time or more clearly.
Great.
Yeah. So I think that what I'm trying to get at is if Aaron is right that there's only really so much statistical information to be had there that's reliable, like when have we just tapped out the methods in imaging, right? Like, and I just think that there is really a ceiling to what in an information theory way of framing it, you can get in principle. So I kind of tend to think of the more sophisticated stuff personally as really creative thought exercises first and foremost, honestly. Whether or not they're really teaching me properties of the system or mechanics I should take to the bank, to me always implies you need another method, period. Like I can't get past the data at that point. So that's for me what usually motivates getting off and doing a brainstem study or something, or looking at comparative literature. I just think you can't get around it.
That makes a lot of sense.
Then take it out of sample and send it more.
Yeah, and so Brian's questions about, well, taking it out of sample and if it's noise, it shouldn't work. So we were talking with briefly Mark Lieberman's group over at Penn, and they were sharing some data they were fitting to the frontotemporal dementia patients. He just pointed out this story I hear over and over when I talk to people that start applying machine learning, which was they had this really good looking linguistic prediction in their data. And Then they realized, well, wait a minute, the appropriate control is just to look at the background noise in the room, and found out they could predict the language information just as well from the background noise in the room, because it was about where the person was sitting, not the content of the language. So you see versions of this problem over and over and over in the literature that are kind of untested because, I don't know, you don't get the right reviewers, like they're complicated, it's easy to miss certain details. But the people who train these methods can share example after example after example of that happening. And so, yeah, it really worries me that, you just need to see more robust data sample. And then even if you're training within your sample, like, look, you want the prediction to work, and it might work, and you cheated in a way you didn't realize, and you just didn't think of the right controls. And it just happens all the time.
Isn't this an issue that the replication movement could help address potentially?
Sure. And Amy, I'm a little maybe cynical beyond my years again, or cynical again, or whatever. But like, what's the NIH still going to get you to fund at a study section? The significant new latest greatest thing. So I think one way I'm trying to solve that problem in my science is we're fond of building designs that let us test the thing we did before while doing the new thing.
Mhm.
And when we pre-register, we've already like preordained we're going to try to do that. So that's been helping us recently, but it still, feels to me like it's an uphill swim a lot of the time. Upstream and uphill. Sorry, I confounded the.
So my other question was a bit of a change of topic, back to the sample size issue. I mean,
Yeah.
so practically, as we've talked about before, a lot of us are plagued by small samples. Or even sometimes we would love to be able to do single case series type analyses. And Solène and I were talking
Yep.
about this earlier, where you can, and so, John, I don't know if this is a question or just sort of a suggestion that we bear this in mind as we go forward working together. That's always going to keep coming up, sort of what's the smallest sample we can do these techniques with and have reliability. Which of these techniques are appropriate for use with single subjects? And
within subject analysis, I would be very interested. We heard in Argis Hillis talk that she presented a few single subject connectivity analysis, and I don't know what the requirements or what's the best way to approach it.
Yeah, and so well, we can always say a lot there, and I'll have recommendations. I mean, I think I now tip my hand that in the functional effective space, I think Gimme USEM, the very verbose but very powerful method, is underutilized. It has very good group to individual profiling features and is very sensitive to variances of both levels.
Can you say it again? What was the method?
The initial, it's Gimme, GIMME.
Yeah.
Oh, yeah, you mentioned that.
And then USEM, USEM, yeah.
Right, okay.
And I'm happy to talk more specifically about use cases there when the time comes. I think in the structure, this is just a personal inclination from the last few years. I am pretty comfortable saying that stroke's going to blow up brain anatomical connections. We're going to see it in the data. We're probably going to see that track with the function in the ways we just discussed. And then kind of like Laurel is getting at, like the closer you are to like corticospinal tract and like, homotopic direct connections you can get in your hypothesis testing, the easier it is to motivate smaller samples work, in my opinion. After that, it gets harder, just exponentially, and you have to be careful. And I think that's where people get into a lot of trouble with small samples.
We should look at that. Give me use some method. Maybe do a journal article on that, John.
I think it's slightly different. I mean, I don't know from a connectivity perspective, but from a statistical analysis, it's not the same thing to do on small samples and within a subject.
Right.
Because generalizing over a small sample is something. It's not the same to really look at a single case. So that if I would rather use a single case than generalizing over a small sample, but yeah,
and I think
I don't know, but
I'm sorry, I didn't mean to cut you off there.
No, that's fine.
Well, I think and if we did a journal club, and I know a lot of people in my lab are interested in this area. what I'll be curious about from you all and your samples is the method explicitly uses the within subject variances trying to project up to the group level and then recursively to the individuals is the trick there. However, I mean, for a long time, I've also been of the view of like, why aren't we just doing more model fitting to the single cases? And in what scenarios should we be doing it? And I think probably most of us have encountered the review problem, right? Where it's like, oh, well, like, what do you say about the group, right? So everybody's like always still kind of compulsed to want to do that. And I'm not sure that's always the point, personally. I don't know.
Right. Yeah, just to follow up very quickly on that aspect of within subject analysis, I think more and more we tend to think about how many people show this particular profile. So it's not that it's only a single case, it's more like, yeah, to be able to conclude at the case level, but then to kind of count or like, it's another way of generalizing to the group, like how many people show this profile within a larger group. So it could still be a model fitting at the individual level, but then so making conclusion at the individual level, but then trying to enlarge the conclusion to a subset of people. So I mean that's what I'm interested in general is more kind of an intermediate approach.
Yeah, great.
Okay. Well, if we don't have any more questions or thoughts, then let's thank John again and Apoorva for a wonderful series, and we'll be in touch with all you guys. Thank you so much.
Yeah, thank you all for listening and interacting. You know where to find us. I hope you all have the rest of the day and good weekend, okay?